walk into a hospital or maybe just think about a blood drive you've seen, or even, the bigger picture, someone getting a life-saving organ transplant.
Mm,
You're right up against one of the most fundamental and honestly one of the trickiest biological systems we have.
mm,
The ability to tell self from non-self.
right. It's the core of our immune defense. It protects us, keeps pathogens out. But when we try to step in medically, like with transfusions or transplants, that same protective system, it becomes the major hurdle.
Exactly. It's this built-in paradox. We've got this fantastic set of notes here from a physiology text that really pulls back the curtain on this whole thing, the really intricate immune reactions, specifically tied to blood and tissue.
Yeah, this material doesn't just skim the surface. It gets right into the mechanics, the how and the why these reactions happen at a molecular level. It's like a blueprint for understanding it all.
Okay, so that's our mission for this deep dive. We're going to unpack this material piece by piece. We want to understand the specific antigens, the antibodies involved, why these reactions actually happen, look closely at the big systems like OAB and RH.
And then tackle the challenges of transplantation too, right?
Yeah.
The HLA system and how we try to manage those immune responses.
Absolutely. The goal here is to go deep, cover every crucial detail laid out in these notes so you can really get a handle on this, understand it like the pros do. We want you to master this complex topic.
It's about building that solid foundational knowledge of the biology dictating success or failure in these critical procedures.
Okay, let's get into it. Starting with blood. The notes point out that the very first attempts at blood transfusions, well, they often ended badly.
Yeah, that's putting it mildly. Historically, it was often disastrous. Early physicians trying this saw these severe, sometimes immediate reactions. The recipient's blood would clump up, that's agglutination, and then the cells would burst hemolysis.
Not just alarming, but often fatal, as the notes say.
Absolutely fatal in many cases. It starkly showed how vital that self versus non-self recognition is. They thought they were just adding more, red fluid.
But the body saw it differently.
Completely differently, like a foreign invasion. And those tragic early failures, well, they were the push scientists needed to figure out that blood isn't all the same. Different people have different antigenic stuff on their red blood cells and different immune components, antibodies floating in their plasma.
So it's the mismatch, the recipient's antibodies hitting the donor's antigens.
Exactly. That's the trigger for the reaction.
So these antigens, they're like little flags or ID tags on the surface of the red blood cells. Is that a fair way to think about them?
What's a pretty good analogy, yeah.
Yeah.
In this context, antigens are substances on the red cell surface that the immune system can recognize and potentially react against.
And it's not just one or two of these tags. The source material mentions there's quite a number.
Oh, yeah. A surprising number. On human red blood cell membranes, we know of at least 30 common antigens and literally hundreds of rarer ones. Any one of them could theoretically cause an immune reaction.
Hundreds. Okay, so why aren't transfusions even more of a minefield? Why don't we see reactions constantly?
That's a good question. It's because most of those hundreds of antigens are what we call weak antigens. They exist, and they're useful for other things like genetic studies, tracking inheritance, figuring out parentage,
Right.
but they generally don't provoke a strong enough immune response on their own to cause a really severe, life-threatening transfusion reaction.
Okay, so out of all these potential flags, there are a few main culprits. Which ones does the source really focus on for major transfusion reactions?
The notes are clear on this. While many antigens exist, two specific systems are overwhelmingly responsible for the significant, dangerous reactions. That's the OAB system and the RH system. These two are the absolute cornerstones of compatibility testing.
Right, OAB. That's the one most people have heard of. Let's start there.
Okay. The OAB system is all about whether you have or don't have two specific antigens on your red blood cell surfaces. These are the type A antigen and the type B antigen.
And they're also called agglutinogens.
Yes, agglutinogens. Because when they meet the right antibody, they cause agglutination, that clumping of the red cells we talked about.
And whether you have A or B or both or neither, that's inherited,
Yes.
right, down to your genes.
Exactly. You inherit genes that determine which of these agglutinogens your body produces and puts on your red blood cells. That pattern defines your basic OAB blood type.
So, based just on having A or B or not, we get the four main types the source lists in Table 36-1.
Precisely. It breaks down like this. If your red cells have neither A nor B agglutinogen, you're type O. Think of O as the absence of A and B.
Okay,
If you have only the type A agglutinin, you're type A.
got it.
If you have only the type B agglutinogen, you're type B.
Yeah, that makes sense.
And if your red cells have both the A and the B agglutinogens, you're type AB. Simple as that, based on those surface tags.
The notes also dig into the genetics behind the ABO locus, the 3 alleles. That explains the inheritance pattern.
Yeah, the genetics are actually quite neat. At the specific spot on the chromosome that codes for ABO type, the ABO locus, there are 3 versions, or alleles, of the gene possible. IA, IB, and IO. Sometimes it's called A, B, and O. You get one from each parent.
And what do those alleles actually do?
Well, the IO allele is basically inactive. It doesn't lead to making a significant A or B antigen. So if you inherit 2 IOs, you get type O blood, no A or B antigen.
Right.
The IA allele codes for making the type A antigen, and the IB allele codes for making the type B antigen. They're the active ones, you could say.
And how do they interact if you inherit different ones, like A from one parent and O from the other?
Good question. The IO allele is recessive. So if you get IA and IO, your genotype is AO.
Yeah.
The IA wins out and you make the A antigen. You have type A blood. Same for IB and IO, genotype BO. You make the B antigen, type B blood.
Okay, so A and B are dominant over O. What about A and B together?
Ah, that's the interesting part. IA and IB are co-dominant. If you inherit IA from one parent and IB from the other, genotype AB, your body produces both the A and the B antigens. Neither one overrides the other, so you end up with type AB blood.
Co-dominant both show up. Okay, so everyone gets 2 alleles. How many possible genetic combinations are there then?
With those 3 alleles, you can have 6 possible combinations, the genotypes. OO results in type O blood.
Right, no antigens.
AA or AO both result in type A blood because A is dominant over O.
Okay.
BB or BO both result in type B blood for the same reason.
Gotcha.
And then the AB genotype results in type AB blood because of that codominance. So 6 genotypes give rise to the 4 observable blood types, the phenotypes, O, A, B, and AB.
That maps it out perfectly. And the source gives some prevalence numbers too, showing how common each type is.
Yes. For the group they studied, type O was most common, around 47%. Type A was next at 41%. Then type B was less common, about 9%, and type AB was the rarest at only 3%.
So O and A are really frequent, B not so much.
Reflects the allele frequencies in that population, yeah. The O and A genes are just more common than the B gene.
Okay, that covers the antigen, the ID tags on the cells. Now, the other side of the coin for reactions, the antibodies or agglutinins in the plasma.
Right, this is where the immune system's response comes in. Agglutinins are antibodies. But here's the key point, and it feels a bit backwards at first. They develop in your plasma specifically against the OAB agglutinogens that you don't have on your own red cells.
Hold on. So your body makes antibodies against the ID tags and it doesn't possess itself. Like it's saying, okay, I don't have that tag, so anything I see with it must be foreign.
That's exactly it. It's the immune system learning what isn't self. If your red cells lack the A antigen, your immune system develops anti-A agglutinins. If you lack the B antigen, you develop anti-B agglutinins.
Okay, let's apply that to the 4 blood types again. What antibodies would each one have floating around?
Okay, following that rule, type O blood cells have neither A nor B antigens. So their plasma contains both anti-A and anti-B agglutinins.
Makes sense. They lack both, so they make antibodies to both.
Type A blood cells have A, but lack B. So their plasma contains only anti-B agglutinins.
Right.
Type B blood cells have B, but lack A. So their plasma contains only anti-A agglutinins.
Got it.
And type AB blood cells have both A and B antigens. Since they possess both, their plasma develops no anti-A or anti-B agglutinins.
That lays out the compatibility map really clearly. Type O fights A and B. Type A fights B. Type B fights A. And AB fights neither. The source also had that interesting chart, figure 36-1, showing when these antibodies actually appear.
Ah, yes, the agglutinin titer over time. It's fascinating. Right after birth, an infant has almost 0 anti-A or anti-B agglutinins. Production starts somewhere between 2 and 8 months old.
So not right away.
No. Then the levels gradually rise, reaching their peak, the maximum titer, usually around age 8 to 10 years.
Peaking in childhood, that's unexpected.
It is. And after that peak, the levels slowly decline throughout the rest of life. This pattern strongly suggests they aren't just genetically programmed in from birth. Something triggers their development.
So the big question, why are they produced? If you're not born with them, what makes your body start making anti-A or anti-B antibodies later on?
The leading hypothesis, which the source material supports, is environmental exposure. The idea is that small amounts of substances that are chemically similar to A and B antigens enter our bodies constantly.
From where?
Through food, or perhaps more importantly, they're found on the surfaces of common bacteria that live in our gut or that we encounter elsewhere.
Ah, so the immune system sees these A-like or B-like molecules on, say, bacteria, flags them as foreign.
Exactly. Flags them as foreign invaders and mounts an antibody response. And crucially, because these A or B structures aren't on the person's own red cells, the immune system doesn't recognize them as self and goes ahead making antibodies.
That makes sense.
The source reinforces this. It mentions if you deliberately give someone who isn't type A some group A antigen, they mount a typical immune response, making more anti-antibodies. And the fact that newborns don't have these antibodies really supports the idea that the stimulus comes after birth from the environment.
So it's basically the body's normal surveillance system doing its job against bacteria, and it just so happens that some bacterial structures look like the A and B antigens on other people's blood cells.
That's the prevailing theory, yes.
Yeah.
It's an overlap, a consequence of our immune defenses.
Okay, we have the antigens on the cells, the antibodies in the plasma. Now, paint the picture. What actually happens during a transfusion when the wrong types meet? Describe that agglutination process.
This is where it gets dangerous.
Okay.
Let's say you give type A blood with its A antigens to a type B person. That type B person has anti-A antibodies in their plasma.
Right.
Those anti-A antibodies immediately find and bind to the A antigens on the surface of the incoming donor red blood cells.
Acting like little bridges between the donor cells.
Yeah.
Exactly like bridges or glue. Antibody molecules have multiple binding sites. IgG has 2. IgM, which is very effective here, can have up to 10. So a single antibody can grab onto antigens on 2 or more separate red cells,
Linking them together.
linking them together, causing them to stick in clumps. That is agglutination.
And the immediate danger of those clumps.
They're too big to pass through the body's smallest blood vessels, the capillaries. They essentially become tiny blockages, plugging up circulation in tissues and organs all over the body.
That's the clumping, the agglutination. But the notes also talked about what happens next, hemolysis.
Yes, agglutination usually leads to hemolysis, the destruction of those red cells. Over the next few hours or even days, those clumps cells get broken down. Partly it's physical stress trying to squeeze through vessels, but mostly it's the body's cleanup crew, phagocytic white blood cells, especially in the spleen and liver, recognizing these damaged clumps and engulfing them.
Breaking them down and releasing their contents.
Correct. And the main content is hemoglobin. Releasing that hemoglobin into the plasma, that's hemolysis. So often in ABO mismatches, it's agglutination followed by this somewhat slower cell-mediated hemolysis.
But the source mentioned a faster, more direct way cells can be destroyed too, acute hemolysis.
Right. That can happen, though it might be less common in typical ABO issues unless antibody levels are very high. In this acute scenario, the antibodies binding to the red cells trigger something called the complement system.
Ah, the complement cascade part of the innate immune system.
Exactly. When complement gets strongly activated, especially by IgM antibodies, sometimes called hemolysins, because they do this, it forms these protein structures called membrane attack complexes.
Sounds ominous.
They are. They basically punch holes directly into the red blood cell membrane. This disrupts the cell's internal balance, ions rush in, water follows. And the cell swells and bursts right there in the bloodstream. That's immediate intravascular hemolysis.
So destruction can be delayed via cleanup cells or immediate via complement punching holes. Either way, hemoglobin gets released, and it's bad news.
Very bad news. Both pathways have severe consequences, which is why preventing the reaction in the first place is absolutely paramount.
Which brings us logically to blood typing, the essential safety check.
Absolutely non-negotiable. Before any transfusion, you have to determine the ABO type of both the donor and the recipient to make sure they're compatible. That's blood typing, and often cross-matching too.
How's the basic typing done? What's the procedure?
The standard lab method described involves taking a blood sample, spinning it down to separate the red cells from the plasma, and then resuspending the red cells in saline. You then take small amounts of this red cell suspension
Okay.
and mix them with specific testing liquids called anti-sera.
Anti-sera meaning they contain known antibodies, right? Like anti-A or anti-B.
Precisely. You mix 1 drop of the cell suspension with anti-serum containing known anti-A antibodies and another drop with anti-serum containing known anti-B antibodies. You let them sit for a few minutes, then look closely, usually under a microscope, for any signs of clumping agglutination.
So if the person's cells are type A, they have A antigens, they should clump when mixed with the anti-A anti-serum.
Exactly. Agglutination means a reaction happened. The antibody in the test serum found its matching antigen on the red cells. The pattern tells you the blood type.
And the source had that clear table 36.2, showing how to read the results.
Yes, it's straightforward. If the cells are type O, no A or B antigens, they won't clump with either anti-A or anti-B serum. Negative, negative.
Makes sense.
If they're type AA antigens, they will clump with anti-A positive, but not with anti-B negative.
Okay.
If they're type B antigens, they won't clump with anti-A negative, but will clump with anti-B positive.
Right.
And if they're type AB, both A and B antigens, they'll clump with both anti-A and anti-B sera. Positive, positive.
Yeah.
It's a quick, reliable test.
Amazing how that simple test prevents such devastating outcomes. Okay, that's the OAB system covered. The source then flags the other system that's just as critical, the RH system.
Yes, RH is the second major player you absolutely have to consider for transfusion safety. But it works a bit differently from ABO.
What's the main difference?
The biggest one is how the antibodies form. Remember how anti-A and anti-B often just develop naturally after birth?
Yeah, from environmental exposure.
Right. With the RH system, that almost never happens. Spontaneous anti-RH antibodies are very rare. To develop significant anti-RH antibodies, an Rh-negative person generally has to be exposed to Rh-positive red blood cells first.
Exposure being the key, like through a transfusion
Or during pregnancy, which we'll get to. But the point is you usually need that initial contact with the Rh antigen itself.
Okay, so what are the Rh antigens? Are there just two, like A and B?
It's actually a bit more complex. The source mentions 6 common types of Rh antigens or Rh factors. C, D, E, and their alternative forms, little c, little d, and little e. Each person inherits one from each pair, so you have either big C or little c, big D or little d, big E or little e.
So you end up with a combination, like C, D, E, maybe.
Exactly. But among those 6, one stands out as being much more important clinically.
That would be the D antigen.
The D antigen, yes.
Yeah.
It's the most prevalent in the population, and critically, it's much more energetic than the others. That means it's far more likely to provoke a strong immune response if introduced into someone who doesn't have it.
And whether you have this D antigen or not is the basis for being called Rh positive or Rh negative.
That's the standard classification, yes. If your red cells have the type D antigen, you're Rh positive. If you lack the type D antigen, you're Rh negative. Now, the source does add that the other Rh antigens, the CE ones, can sometimes cause transfusion reactions in Rh-negative people. But these reactions are usually much milder than those involving the D antigen or an ABO mismatch.
Got it. D is the main player. And the source gives some stats on how common Rh positive versus negative is.
Yeah, it varies a bit by population. Among white individuals, about 85% are Rh positive, 15% H negative. It's higher in other groups, around 95% positive in American blacks, almost 100% in African blacks, and over 95% in Native Americans and Asians. Globally, it's estimated around 95% of people are Rh positive.
So Rh negative is relatively uncommon overall. Okay, let's talk about the immune response. How does that exposure actually trigger antibody production in an Rh negative person?
So an Rh negative person gets exposed to Rh positive red cells carrying the D antigen. Their immune system sees D as foreign and starts making anti-Rh antibodies, specifically anti-D. But here's a key difference from ABO. It's slow. The source says it takes about 2 to 4 months after that first exposure for the antibody levels to peak.
2 to 4 months. That's a long lag time compared to the potential immediacy of ABO.
A very different timescale.
Mm-hmm.
And how strong that response is, the final antibody titer, varies a lot from person to person. But if that person gets exposed again to the Rh factor,
the response is stronger.
Much stronger and faster. Repeated exposures lead to greater sensitization. The immune system gets primed.
Okay, so what does this mean practically for transfusion reactions? What happens the first time an Rh negative person gets Rh positive blood versus the second time?
This difference is absolutely critical. On the first exposure, say, an Rh negative person mistakenly gets a unit of Rh positive blood. There's usually no immediate reaction. The Rh antibodies just aren't there yet in significant amounts.
Okay.
Over the following 2 to 4 weeks, the antibodies gradually develop. As their levels rise, they start to agglutinate any of those transfused Rh-positive cells that are still hanging around in the circulation.
And then those clumped cells get cleared out?
Yes, those agglutinated cells are then destroyed, mostly by the tissue macrophage system, similar to the delayed ABO hemolysis. So the first mismatched Rh transfusion typically results in a delayed reaction, and it's often quite mild.
If that same person, who is now sensitized, gets Rh-positive blood again,
now you have a problem. Because their immune system is primed, the response to the second exposure is massively amplified. It can be immediate and severe, potentially just as bad as a major ABO reaction. Immunological memory kicks in, churning out high levels of anti-Rh antibodies very quickly.
So the first exposure is like the immune system doing reconnaissance, and the second time it launches a full-scale attack.
That's a great way to put it. It's ready to go much faster and much harder the second time around.
This whole Rh exposure scenario is also the direct cause of that serious condition in newborns, right? Erythroblastosis fetalis.
Exactly. Erythroblastosis fetalis, or the hemolytic disease of the newborn. It's a disease where the baby's red blood cells are attacked and destroyed by antibodies from the mother that have crossed the placenta.
And the source lays out the classic setup for this.
The classic, most common cause is an Rh-negative mother carrying an Rh-positive fetus. The father is Rh positive, so the baby inherits the Rh positive trait.
Okay, Rh negative mom, Rh positive baby. How does the mother actually get exposed to the baby's Rh positive cells? She's Rh negative herself.
Small amounts of fetal red blood cells can leak across the placenta into the mother's circulation during the pregnancy. This is particularly common during delivery, when the placenta separates, but can happen earlier too, maybe due to tiny bleeds.
Ah, okay.
Since the mother is Rh negative, her immune system sees these fetal Rh positive cells as foreign and starts producing anti-Rh antibodies, the same sensitization process we just discussed for transfusions.
And then her antibodies can get back across the placenta to the baby.
That's the problem. Once the mother has developed enough anti-Rh antibodies, these antibodies, being typically IgG class, can cross the placental barrier into the fetal bloodstream. There, they attack the baby's Rh-positive red blood cells.
So the mother's immune system, trying to protect her from what it sees as foreign, ends up harming the baby. That self-non-self paradox again.
A very clear and sometimes tragic example of it, yes.
What's the risk pattern? Is the first Rh-positive baby usually affected?
Generally, no. The source confirms the risk for the first Rh-positive baby is low. The mother usually doesn't build up a high enough level of antibodies during that first pregnancy to cause major problems, but that first pregnancy often sensitizes her.
Making later pregnancies riskier.
Exactly. With subsequent Rh-positive pregnancies, her immune system is primed. Exposure happens again, antibody levels rise faster and higher. The source quotes figures like maybe 3% of second Rh-positive babies showing effects, 10% of third, and the risk keeps climbing.
So what actually happens to the fetus or newborn when these maternal antibodies attack?
The antibodies cause agglutination and hemolysis of the fetal red cells. This leads to anemia in the fetus. As the hemoglobin breaks down, bilirubin levels rise, but the fetal liver isn't fully equipped to handle it. After birth, the mother's antibodies are still circulating in the baby for a month or two, continuing the destruction.
So the baby is born anemic and the attack continues. How does the baby's body try to fight back?
The baby's own blood-forming tissues, the bone marrow, but also significantly the liver and spleen go into overdrive. They try desperately to produce new red blood cells to replace the ones being destroyed. These organs often become very enlarged.
And this rapid production pushes out immature cells.
Yes. Lots of immature nucleated red blood cell precursors get released into the circulation. That's actually where the name comes from. Erythroblastosis fetalis refers to the presence of these blastic immature red cells.
What are the most severe potential outcomes if it's bad and not treated?
The anemia itself can be fatal, but the other huge danger is the high level of bilirubin. Bilirubin is toxic to nerve cells. If levels get too high, it can deposit in parts of the brain, especially the basal ganglia, causing permanent brain damage. This condition is called kernicterus.
Kernicterus. That sounds devastating. Permanent brain damage.
Yes, it can lead to permanent mental impairment, problems with movement control, conditions like cerebral palsy. Preventing kernicterus is absolutely critical.
So what's the treatment for a baby born with severe erythroblastosis fetalis?
The main treatment described is exchange transfusion. It's a procedure where the baby's Rh-positive blood is gradually removed while simultaneously being replaced with compatible Rh-negative blood.
Literally swapping out the blood. Why Rh-negative blood?
Because the Rh-negative blood doesn't have the D antigen, so the mother's anti-Rh antibodies still circulating in the baby have nothing to attack.
Ah, okay. And what does the exchange transfusion achieve?
It's usually done slowly, over maybe 90 minutes or more, and might need repeating. It does two crucial things. First, it provides healthy red cells, helping correct the dangerous anemia. Second, and very importantly, it removes the baby's blood that's full of bilirubin and replaces it, drastically lowering the bilirubin level and preventing it from reaching toxic levels that cause kernicterus.
And over time, the baby will naturally replace the transfused Rh-negative cells with its own Rh-positive ones.
Yes, that process takes about 6 weeks or so. By then, the maternal antibodies that cross the placenta should have naturally degraded and disappeared from the baby's system. If the baby got through that critical early period without brain damage, their own Rh-positive cells can then survive normally.
It's a complex intervention, but has prevention become possible? The source mentioned progress there.
Oh, absolutely. Prevention has been a massive success story in medicine since the 1970s. The source definitely highlights this.
How is it prevented?
The key is administering something called Rh immunoglobulin. It's basically a concentrated dose of anti-D antibodies.
Wait, you give the mother anti-D antibodies to stop her from making anti-D antibodies? That sounds backwards.
It does seem paradoxical, doesn't it? The exact mechanism isn't perfectly understood, but it's incredibly effective. The source suggests a couple of possibilities. One idea is that the injected antibodies somehow interfere with the mother's own immune cells, her B lymphocytes, suppressing their ability to get activated and produce their own anti-D antibodies.
Like it short-circuits her response.
Sort of. Another strong possibility is that the injected anti-D antibodies quickly find and bind to any fetal Rh-positive cells that have leaped into the mother's circulation. By coating these cells, they essentially hide the D antigen from the mother's immune system, preventing it from recognizing the cells as foreign in the first place.
So either block the response or hide the target. When is this Rh immunoglobulin given?
It's typically given to Rh-negative mothers around 28 to 30 weeks of pregnancy as a precaution, and then definitely again shortly after delivery if the baby is confirmed to be Rh-positive. It's also given after events like miscarriage or amniocentesis, where fetal cells might have entered the mother's system.
And this has drastically cut down on erythroblastosis fetalis.
Dramatically. It's turned a relatively common and often tragic disease into a rare one, at least where good prenatal care, including this prevention, is available. A real triumph.
Truly remarkable. Okay, before we shift fully to tissues, let's clarify one point from the transfusion reaction section. The source noted that usually it's the donor cells that get attacked, not the recipients. Why is that the case?
It boils down to simple dilution, really. When you give a transfusion, it's usually, a unit or two? Maybe 300, 500 milliliters of blood?
Right.
The recipient has a much larger total blood volume, several liters. So the plasma portion of the donor blood, which contains the donor's antibodies, gets instantly diluted in the recipient's large volume. The concentration of those donor antibodies becomes very low.
Too low to cause much trouble for the recipient's own cells.
Usually, yes.
Uh-huh.
Too low to cause significant agglutination of the recipient's vast number of red cells. But flip it around. The recipient's own antibodies, already present in their plasma, don't get significantly diluted by the relatively small volume of incoming donor blood.
So the recipient's antibody concentration stays high relative to the number of incoming donor cells they can attack.
Exactly. The recipient's immune system has the numerical advantage, you could say. Their antibodies are concentrated enough to effectively attack the antigens on the smaller population of transfused donor cells. That's why the reaction primarily targets the donor cells.
That makes perfect sense. Now, after hemolysis happens and hemoglobin is released, what are the main downstream consequences mentioned?
Okay, so hemoglobin spills into the plasma. Phagocytic cells, especially in the liver and spleen, grab this free hemoglobin and break it down into bilirubin. The liver then normally processes this bilirubin and excretes it into the bile.
And if there's too much bilirubin too quickly?
Then the liver gets overwhelmed. Bilirubin levels build up in the blood, causing jaundice, that yellowing of the skin and eyes. The source mentions that in an adult with a healthy liver, you'd need quite a bit of hemolysis, maybe over 400 milliliters of blood destroyed in a day, for jaundice to become obvious.
Jaundice is a serious sign, but the source pointed to an even more lethal outcome of severe reactions.
Yes, acute kidney failure. This is highlighted as one of the most dangerous possibilities. It can develop quickly after a severe reaction and is often fatal without immediate treatment like dialysis.
How does destroying red blood cells cascade into shoving down the kidneys?
It's a multi-pronged attack on kidney function, triggered by the systemic effects of the reaction. The source breaks it down into three main causes working together.
Okay, what's the first?
First, renal vasoconstriction. The antigen-antibody reaction itself, plus substances released from the damaged red cells, causes the blood vessels within the kidneys to clamp down forcefully. This drastically reduces blood flow through the kidneys.
Less blood flow impairs function. What's number two?
Second is circulatory shock. The whole process, losing red cells, toxic substances floating around, the intense immune reaction can cause a dramatic drop in the person's overall blood pressure. This circulatory shock further starves the kidneys and other organs of blood flow and leads to very low urine output.
Okay, low flow from constriction plus low pressure from shock. What's the third factor?
The third is a direct physical blockage. When massive amounts of hemoglobin are released, more than the body's carrier proteins like haptoglobin can handle, the excess free hemoglobin gets filtered through the kidneys glomeruli into the renal tubules, where urine is made. Normally, water is reabsorbed from these tubules to concentrate urine. But if there's a ton of hemoglobin in there as water leaves, the hemoglobin gets more and more concentrated until it actually precipitates. It turns into solid casts that physically plug up the tubules.
So the kidney's plumbing gets clogged with hemoglobin sludge.
Essentially, yes. So you've got this devastating combination: severely constricted blood vessels cutting off supply, circulatory shock dropping pressure, and physical blockage of the tubules. Together, these lead to acute renal shutdown.
And if that shutdown is complete and isn't treated,
The source says it's usually fatal within about 7 to 12 days unless the person can be put on an artificial kidney, dialysis, to take over the kidney's filtering function.
that really drives home the potential severity. An immune reaction starting with blood cells can spiral into total kidney failure. It highlights the interconnectedness and the danger.
Absolutely. It shows how localized immune events can have profound systemic consequences.
All right, we've thoroughly explored blood. The source material then broadens the scope to transplanting tissues and organs. Is the basic challenge the same?
Fundamentally, yes. It's still that self versus non-self recognition by the immune system. The source notes that many of the antigens found on red blood cells are also present on other body cells. Plus, tissues and organs have additional, unique antigens of their own. So when you transplant foreign cells or a whole organ, the recipient's immune system views it as foreign.
And attacks it, just like it would attack bacteria or mismatched blood.
That's the core issue, yes. The
Yeah.
immune system mounts a response aimed at rejecting and destroying the foreign tissue. This immune rejection is the biggest obstacle in transplantation.
The source defines different categories of transplants or grafts. Can you quickly run through those? Autograft, isograft.
Sure. An autograft is tissue moved from one part of a person's body to another part of the same person, like taking skin from your thigh to cover a burn on your arm. Immune rejection isn't a factor because it's all self. An isograft is a transplant between genetically identical individuals, specifically identical twins. Since their genes, and therefore their antigens, are the same, rejection is usually not an issue here either.
Right.
An allograft is a transplant between two different individuals of the same species who are not identical twins. This is the most common scenario in human organ transplants: a kidney from a sibling, a heart from an unrelated deceased donor. Immune rejection is the major challenge with allografts.
Got it. And the last one, xenograft.
A xenograft is a transplant across species barriers. For example, tissue from a pig to a human, or between different animal species. Here, the antigenic differences are huge, and the immune rejection is typically very strong and rapid.
So, summarizing the rejection risk, auto and isografts are safe immunologically. Allografts are the main challenge we face in human medicine. Xenografts are extremely difficult.
That's a good summary. The source says autografts and isografts usually survive indefinitely if the blood supply is good. Xenografts almost always face rapid rejection unless extreme measures are taken. Allografts are where success is possible, but requires managing that immune response.
And the source lists common allografts: skin, kidney, heart, liver, lung, bone marrow. Mentioning success rates for kidneys can stretch 5-15 years or more with good management.
Right, which shows that while rejection is tough, we've developed strategies to deal with it, allowing for long-term graft survival in many cases.
Which leads us straight into how do we try to overcome these immune reactions in allografts?
This has been a huge area of medical research, obviously. The key strategies involve trying to minimize the antigenic differences between donor and recipient and suppressing the recipient's immune response.
Okay, minimizing differences. Beyond basic ABAR matching, the source emphasizes tissue typing, focusing on something called the HLA complex.
Exactly. While ABO compatibility is important for transplants too, the most critical antigens for triggering graft rejection are the human leukocyte antigens, the HLA system.
HLA. Why is the system so problematic for matching?
The sheer diversity is incredible. These HLA antigens are on the surface of most tissue cells, especially white blood cells, leukocytes. The source states each person inherits and expresses 6 main HLA antigens. But in the human population as a whole, there are about 150 different versions of these HLA antigens known.
Whoa, 6 slots, but 150 options to fill them. The number of combinations must be astronomical.
Truly astronomical. The notes calculate it's over 1 trillion possible combinations. This means, unless you're an identical twin, the chance of finding someone else with the exact same 6 HLA antigens is practically zero. And the immune system can potentially react against any HLA antigen difference between the donor and recipient.
How on earth do they even attempt to type and match something that complex?
It's done through detailed laboratory testing called tissue typing, usually using lymphocytes from the blood. The process is similar in principle to blood typing, but much more extensive. The lymphocytes are mixed with a large panel of different anti-sera, each containing antibodies known to react with specific HLA types. Complement is added. If an antibody finds its matching HLA antigen on the lymphocyte, it binds, activates complement, and damages the cell membrane.
And they detect that damage.
Yes, often using dyes that can only enter damaged cells. By seeing which anti-sera cause damage, they can map out which HLA antigens the person has.
A complex process. So, how important is getting a good match? Is a perfect 6-antigen match essential?
A perfect match is the ideal, of course, and gives the best chance of success with minimal rejection. But the source notes that not all HLA antigens trigger equally strong reactions. Some mismatches are tolerated better than others, especially with modern immunosuppression. So while a perfect match isn't always absolutely required, the better the match, the lower the risk of rejection.
And matching is easier between relatives.
Much easier. Siblings have a 1 in 4 chance of being a perfect HLA match. Parents and children always share half their HLA antigens. This genetic relatedness is why close relatives are often preferred donors. The HLA similarity significantly improves outcomes. And identical twins, again, are a perfect match.
Okay, so matching helps, but finding a perfect match is rare outside of twins. What's the other main strategy? Suppressing the immune system.
Yes, that's the cornerstone of modern transplantation. If you can dampen the recipient's immune response, you can prevent it, or at least lessen its ability, to attack and reject the foreign graft.
But that sounds risky. Doesn't suppressing the immune system leave the patient vulnerable?
That's the critical trade-off, and the source is very clear about this. Yes, effective immune suppression prevents rejection, but it comes at a cost. The patient becomes much more susceptible to infections, bacteria, viruses, fungi, which can become severe or even fatal.
And there was another risk mentioned besides infection.
Yes, a significantly increased risk of developing cancer. The thinking is that our immune system normally performs surveillance, finding and destroying early cancer cells. When you suppress that surveillance, cancers have a better chance of taking hold and growing.
That's a really tough balancing act for doctors and patients. Which parts of the immune system are most important to suppress for preventing graft rejection?
The source specifically points to T cells. While antibodies, made by B cells, play a role, the T lymphocytes are considered the primary mediators of the direct cell killing that leads to graft destruction. So therapies often focus heavily on suppressing T cell function.
What kinds of drugs or therapies are used to achieve this T cell suppression, according to the source?
Several classes of agents are mentioned. First, glucocorticoid hormones like prednisone or similar drugs. They work broadly by inhibiting genes that code for immune signaling molecules called cytokines, especially interleukin-2 or IL-2.
And IL-2 is key for T cells.
Absolutely key. IL-2 drives T cell proliferation and activation. So blocking it significantly dampens the T cell response and antibody formation too. Second, there are drugs generally toxic to the lymphoid system, like azathioprine. These hit rapidly dividing cells, including lymphocytes, thus blocking the production of both T cells and antibodies. They're less specific, though.
Okay, more targeted options.
Yes. Thirdly, and very important clinically, are drugs like cyclosporine and tacrolimus. These specifically inhibit the function of T helper cells. T helper cells are like the conductors of the immune orchestra, especially for T cell-mediated rejection. These drugs are valuable because they block that pathway effectively, often without depressing all other immune functions quite as severely.
That sounds like a better balance.
Potentially, yes. And fourth, the source mentions immunosuppressive antibody therapy. This uses engineered antibodies designed to target specific immune cells or molecules. Examples include antibodies that kill lymphocytes directly, antilymphocyte globulin, or antibodies that block the IL-2 receptor on T cells, preventing them from getting the activation signal.
So a whole toolkit of drugs targeting different parts of the immune response, often with a focus on T cells.
Exactly. And the source really credits the development and refinement of these immunosuppressive drugs for the success we see in organ transplantation today. They've made procedures that were once experimental much more routine and successful.
So the modern approach is all about using these tools carefully, trying to find that sweet spot, enough suppression to protect the graft, but not so much that the patient is overwhelmed by side effects like infection or cancer.
That's the constant challenge, yes. Balancing graft survival with patient safety. It requires ongoing monitoring and adjustment of the immunosuppressive regimen for each individual patient.
Wow. We've covered a huge amount of ground. From, the basics of A reacting with anti-A in a blood test, all the way to the incredible complexity of HLA matching and immune suppression for organ transplants.
It really underscores how sophisticated and sometimes problematic our immune system's ability to distinguish self from non-self truly is. That fundamental process underlies everything we've discussed.
We saw how simple ABO or RH mismatches can cascade into agglutination, hemolysis, even kidney failure.
Yeah.
And then how the much more intricate HLA system demands complex tissue typing and powerful drugs to make life-saving transplants possible. Hopefully, this deep dive, drawing on all the details in our source material, has provided that foundational understanding you were looking for.
Yeah, the aim was to unpack the specific antigens, the antibodies, the reaction processes, the classification systems like ABO, RH, HLA, and the medical strategies involved, hopefully bringing you closer to that expert-level grasp of this vital area of physiology.
Understanding the why behind blood compatibility rules or the necessity of anti-rejection drugs really gives you a new appreciation for these medical interventions.
It absolutely does. And, it leaves you with a pretty profound thought about our own biology. Think about it. The very system that's absolutely essential for protecting us from disease, this powerful immune surveillance that identifies and destroys anything foreign, is the exact same system we have to carefully navigate, manipulate, and often actively suppress, just to save lives through things like blood transfusions and organ transplants.
That's a fascinating point. The defense mechanism becomes the obstacle.
Right. What does that inherent trade-off, that tension between essential protection and the need for medical intervention, tell us about the fundamental design principles, maybe even the compromises built into our own bodies?
Something definitely worth pondering. Thanks for joining us on this deep dive.